In this next video clip, we're going to begin to talk about some of the environmental factors that influence microbes. So as I mentioned earlier, and as you no doubt have read in your textbook by now, microorganisms are adapted to many different environments, and those environments that they're adapted to, those are going to influence their metabolism. So metabolism, all these catabolic and anabolic reactions that are necessary for harnessing energy and then using that energy to build all of your cell parts and maintain yourself as a cell or an organism, those things have to operate optimally in the environment where an organism lives if the organism is going to be a good surviving member of that environment. So these various factors that we get exposed to certainly influence survival. All right, and as I've mentioned also, microorganisms are adapted to many different environments. Different microorganisms are adapted to different environments. All right, so what are some of these environmental factors that will impact the growth of microorganisms? So temperature will be one thing we'll take a look at. Oxygen requirements. Not all microorganisms require oxygen for survival, and in fact, for many microorganisms, oxygen is actually toxic, as we'll see, believe it or not. pH, are you adapted to acidic pHs, neutral pHs, or alkaline? Osmotic pressure, so that gets into solute concentrations and what kind of fluids are isotonic, hypertonic, or hypotonic for you, because that's going to influence the movement of water across your cell membrane. And if you're a little microbial cell, you don't want to burst and you don't want to shrivel up, so you've got to be adapted to a proper solute concentration. Radiation tolerance. And here we're really going to be thinking about light as a type of radiation. We're used to thinking of radiation as something dangerous for us, but light, visible light, is actually a type of radiation as well. And then the influence of other organisms in your surroundings. What kind of impact do those have on your survival as a microorganism? All right, and as we're going through this, keep in mind not only thinking about these organisms and how they adapt to, and what particular environments they're adapted to, but for culture in a laboratory, if you want to grow an organism in the laboratory, you've got to mimic all of these environmental factors that they're adapted to as well. So that's important for proper diagnosis or research or whatever it is you're trying to do with these little microorganisms in the lab. All right, the first one is temperature. And temperature, all living things have an optimal temperature at which they grow best. Okay, and so here's a little graph over here, and on our y-axis we've got growth rate, and then we've got temperature going from low to high. And now we don't have any temperatures drawn in here because this is reflecting that different kinds of microorganisms on our planet are adapted to different temperatures. But all organisms are going to have some optimal or optimum temperature at which their growth rate is the highest. And what that means is they're reproducing the fastest at that temperature. All right, as you go downward this way to cooler temperatures than the optimum, you'll see the growth rate will drop off. Now, for most microorganisms, that drop off is going to be fairly slow, and it just basically means your metabolism is slowing down. Your metabolism is a sum of a bunch of chemical reactions, and chemical reactions tend to occur more slowly as temperatures get cooler. And eventually you're going to hit what's called the minimum growth temperature. That's the coolest temperature at which an organism can still grow to some degree. So it may be extremely slow, but there will still be some cell reproduction going on at that temperature. Now notice as you go above the optimum temperature, you also reach what we call the maximum growth temperature. And now maximum growth temperature is not the optimum, because it kind of sounds like when you're first listening to it that maximum growth temperature would be the temperature at which growth happens maximally, and that's not true. The maximum growth temperature is the highest temperature at which you get even a little bit of growth. So growth there will still be pretty pathetic, but at some point you do hit the highest temperature where you can get any kind of growth at all for an organism. All right, an example of this is shown on some petri dishes on which bacteria have been streaked out to try to generate colonies. And so this is the same organism, and you can see over here at 37, you've got a pretty high amount of growth. 30 degrees Celsius, you've still got a pretty decent amount of growth, but not as much as at 37 Celsius. And then when you go down to 22 Celsius, you have some growth in the parts of the plate where a lot of bacteria were applied, but you don't see nearly as much as you do on those other two plates. 37 Celsius, keep in mind that's human body temperature. So 30 degrees Celsius is going to be, let's see, 60, 54, about 86 Fahrenheit. And 22 Celsius is kind of like a cool room temperature level. And with a lot of the bacteria that you would grow in an actual microbiology lab where you're interested in bacteria that impact humans and live on or inside the human body, the optimal growth temperature for those types of microorganisms will be human body temperature. If you're adapted to growing on or inside the human body, you're probably going to be optimally adapted to human body temperature. And with those types of organisms, if you shift their temperatures downward, they will still grow, but they're just going to grow at a slower rate. The main thing that temperature impacts is the 3D structure, in addition to the speed of chemical reactions, the 3D structures of biological molecules. So this is important. Proteins, their 3D shape is absolutely critical for them to be able to do their job, whatever it is. You know, if there might be a membrane transport protein that transports glucose across the membrane, it might be an enzyme that carries out a chemical reaction. Whatever it is, that 3D structure is important. When temperatures get too high, proteins tend to unravel. They lose their 3D shape. They're said to denature, is the term for that. Lipids at cold temperatures, your membrane lipids become too rigid. A membrane is supposed to kind of be a fluid, flowing type of structure, believe it or not. It's not supposed to be a rigid structure, and so cold temperatures can impact lipids. And a DNA at really high temperatures, DNA, double-stranded DNA, will actually unwind. And that's not good for an organism either, for that to happen when you don't need it to. And so those are some different ways that temperature will impact biological molecules. And this, the denaturing of proteins, is why you see this very quick drop-off in how well an organism grows as you go above the optimal temperature. Because if all your proteins are nice and folded up optimally at 37 Celsius, let's say here, if you go just a few degrees warmer than that Celsius, they pretty quickly begin to unravel and lose their function. Okay, so microorganisms can actually be categorized according to their temperature ranges for growth, and unfortunately this figure that I pulled out of the textbook doesn't have the labels on here, but take a look at this in your textbook. So this is supposed to be growth over here on the y-axis of the graph, and then these are different temperature ranges, and again, be sure you take a look at this in your textbook. The blue line over here is showing optimal growth for a group of organisms called psychrophiles. So organisms can be categorized according to their temperature preferences, and that's psychro, it's not psycho. Psychrophiles are, and phile means to love, like hydrophilic, water-loving. Psychrophiles are organisms that love cold temperature. So their optimal temperature right in here will be somewhere below 15 degrees Celsius, which is 15 times 2 is 30, 27, 30. That's 59 Fahrenheit. And then for some of them, they go even colder. So there are some psychrophiles that actually thrive on snow and ice. That's where they grow well. All right, your mesophiles. Meso, that prefix means middle. Those are organisms kind of here in green. 20 to 40 degrees Celsius will be the optimal growth temperature. So humans are mesophiles. The pathogens, the pathogenic microbes that we study in this class are going to be mesophiles since they're adapted to the human body. Then this hot pink line represents a class of organisms called the thermophiles, and then over here you have a graph representing hyperthermophiles or extreme thermophiles. So thermophiles thrive at temperatures above 45 degrees Celsius. When you get over here in the hyperthermophile or extreme thermophile category, some of those organisms actually thrive in temperatures over 100 degrees Celsius, which is the boiling point of water. A couple of other special categories I want to point out. Psychrotrophs. So psychrophiles, their optimal growth temperature is below 15 Celsius. Psychrotrophs are organisms that prefer warmer temperatures. They're probably mesophiles, but they can still grow pretty well in the cold. And then among your mesophiles, there are organisms that are called thermoduric. So even though they like middle temperatures, they're durable to higher temperatures, so they can withstand exposure to higher temperatures better than most mesophiles can. All right, so in this PowerPoint, which is posted for you guys on Blackboard, I have an embedded video here you might want to take a look at. Some of you guys may remember this. This is a news story about an outbreak or a contamination of cantaloupes a couple of years ago with listeria bacteria. Listeria monocytogenes, is a type of psychotroph. So that means it's a mesophile. It likes middle range temperatures. Listeria is a human pathogen, so it's going to like 37 degrees Celsius temperatures, but it's a psychotroph, so it can actually still grow in colder temperatures. So unfortunately, this organism can cause a type of food poisoning, and there are outbreaks of this fairly frequently. I'm recording this video in June of 2015, and just a couple of months ago, there was a listeria outbreak in Bluebell ice cream, and all the Bluebell ice cream got pulled off the shelves, which is something more tragic for most of us than having to pull the cantaloupes off of shelves. Although I do like cantaloupe, hopefully you do as well. But anyway, this organism, so with the cantaloupes or with the ice cream, this organism got into it, and unfortunately when that happens, it's still capable of surviving and growing in cold temperatures, and then it can make some people sick. Now fortunately, listeriosis typically only occurs in people with weakened immunity, but that can include older people, young children. If you're pregnant, your immune system is suppressed during your pregnancy, so you have to be careful about that as well. You may have heard that pregnant women should not eat uncooked cold cuts like hot dogs, things like that, and the reason for that is because sometimes hot dogs can be contaminated with listeria. If you cook them, it gets killed, but if you eat them cold, you're pregnant, your immune system is lower, you're more susceptible to a listeria infection. This organism too, Staphylococcus aureus, we'll hear about that organism a lot during this semester. That is an organism that causes staph infections, and it's a pretty mean little critter. It comes up a lot during the semester, and unfortunately it's one of these things that can also grow at colder temperatures. It's also a psychotroph. All right, the next physical requirement I want to discuss is the oxygen gas requirements of organisms. Okay, so not all organisms require oxygen gas to grow like we do. Now, it is true that O2, oxygen gas, is required by many microbes for survival, and the ones who do need it, they need it for the same reason we do. They need it for cellular respiration, which we will talk about more as we get into Chapter 7. But this is an overall summary reaction of cellular respiration. These are your inputs over here and your outputs over here. And again, we'll talk more about this when we get into Chapter 7. But this is glucose, C6H12O6. So you start cellular respiration with a glucose monosaccharide or sugar. You also need 6 oxygen gas molecules as inputs into cellular respiration for every glucose molecule you start off with. And then your outputs over here are 6 carbon dioxides. So this glucose is going to get broken up. It's going to get broken down, and as it gets broken apart, remember how we talked about how the covalent chemical bonds in a molecule have energy stored in them, and when you break those bonds, energy gets released. So in cellular respiration, you're breaking apart glucose, and the idea there is to harness the energy and store it in the form of ATP, those fuel molecules that cells need for survival. So as glucose gets broken apart, all those carbons in a glucose, they end up over here in carbon dioxide molecules. You got 6 carbons in a glucose, and you end up with 6 carbon dioxide molecules as you finish cellular respiration. 6 water molecules are outputs of cellular respiration. You generate ATP, and again, we're going to see that as more when we get into Chapter 7. And then heat is also a byproduct of cellular respiration. But anyway, for every glucose molecule, you got 6 oxygen gas inputs, and this is why we need oxygen gas, and this is why many other organisms on the planet need oxygen gas as well. Critical for cellular respiration. And we can't survive without oxygen gas because we need it to run cellular respiration, and quite a few microbes can't survive without it either. So what's the problem? The problem is oxygen gas sounds wonderful. What's the issue? Oxygen gas pretty quickly converts inside cells to other forms, other oxygen-containing molecules that are very toxic and chemically reactive. And those include things that are called singlet oxygen-free radicals, superoxide ion, where you have an O2 but it has an extra electron, so it has a negative charge. It can react and become hydrogen peroxide, H2O2, hydroxyl radicals. That's an oxygen and a hydrogen atom bonded together with an extra electron, a negative charge. So all of these things are generated just by normal natural chemical reactions all the time that take place in cells exposed to O2. And all of these things are very highly chemically reactive. Now over here it's showing you in this figure, well, what are some of the things that cause or trigger these O2 molecules to react with other things in the cell and produce these toxic oxygen-containing compounds. So radiation exposure, UV light from the sun in the human body. If you're a smoker, you have more of these toxic oxygen-containing molecules produced, air pollution, etc. But the biggest problem is these will react with things like your DNA. They can damage your DNA. And same thing in a microorganism. Your DNA is your genetic blueprints. If you lose your genetic blueprints, you're pretty quickly not going to be able to build all your cell parts and operate properly, and you're going to die if you accumulate enough of that damage. So these oxygen-free radicals are not good news for living things. All right. Now you may have heard of antioxidants before. You know how they tell you to make sure you eat foods that are rich in antioxidants. So I know things like blueberries come up as pomegranates, stuff like that, all rich in antioxidants. And what antioxidants are, they are compounds, molecules that will convert these nasty oxygen-containing compounds back to a safe molecule that's not chemically reactive. So those are called antioxidants. And they will help, if you have more antioxidants around in your cells, it helps limit DNA damage that takes place. So you'll hear about those a lot of times because DNA damage can lead to cancer. So if you consume more antioxidants in your diet, it may limit some of the DNA damage that takes place in your body, and it might help lower your risk for cancer. Okay, so all living things that live around oxygen gas have to have ways of destroying these naughty oxygen-containing compounds. And some examples are they do this through the use of enzymes. So there are various enzymes. Enzymes are we're going to talk more about enzymes in Chapter 7, but they're proteins, usually, almost always. And these proteins function basically like little machines that carry out chemical reactions. They force them to happen. And enzyme names usually have ASE on the end. So one enzyme is called superoxide dismutase. So a lot of living things that grow and survive in the presence of oxygen gas have these enzymes. And so if you have these oxygen gas molecules, superoxide ions with the extra negative charge, what this enzyme will do is it'll take 2 of those and combine them with 2 hydrogen ions that are floating around in the cell fluids, and creates H2O2, that's hydrogen peroxide, plus an oxygen gas molecule. So this is the friendly oxygen gas molecule that we can use for cellular respiration. So superoxide dismutase is an enzyme that makes that chemical reaction happen. These are your inputs. These are your outputs. Now, hydrogen peroxide is itself a very chemically reactive molecule, so it has to be inactivated as well. And this gets generated in any cell that's exposed to oxygen. This hydrogen peroxide molecules just naturally get produced all the time. And so there are a couple of different kinds of enzymes that organisms can have that will inactivate hydrogen peroxide. One of those enzymes is called catalase, and so it will take these and convert it to water and oxygen gas, both of which are safe molecules. And then peroxidase enzymes perform a different kind of chemical reaction where they take H2O2 molecules and just convert them to water. So a couple of different ways that organisms can inactivate H2O2. Now, the presence of catalase is actually the basis for a test that's used in microbiology laboratories. So what you can do, we have catalase enzymes in our cells, and so do many bacteria, but not all of them. So what you can do is you can smear some bacteria, like some growth from a culture, on a glass slide, and just drip some H2O2 on the bacteria. If those bacteria have catalase enzymes, that H2O2 that you're dripping on them will get converted to water and oxygen gas. So it'll bubble. If they have catalase enzymes, you'll see these oxygen gas bubbles being given off, and that lets you know that that organism is positive in the catalase test. You guys, if your mom ever put hydrogen peroxide on a cut, you know that it bubbles, and the reason it's bubbling is because the catalase enzymes in your cells are converting that hydrogen peroxide very quickly to water and oxygen gas. That also means that your cells are inactivating the hydrogen peroxide pretty quickly, so that hydrogen peroxide is probably not doing a whole lot for you, at least on a long-term basis. It is helping to remove or kill some of the bacteria that are there, but not all of them, because your own cells are going to inactivate it pretty quickly. All right, so microorganisms can be categorized according to their oxygen requirements. So many microorganisms are what we call obligate aerobes. Aero in biology generally refers to O2 or oxygen gas. Obligate, obligatory, you've got to do it. These are organisms that are like us. We're obligate aerobes. They can only survive when oxygen gas is available. A couple of pathogens associated with that, Pseudomonas aeruginosa and Mycobacterium tuberculosis. Those are both obligate aerobes. This is an image of an ulcer on the bottom of someone's foot, and this is probably a diabetic patient, and this is a Pseudomonas aeruginosa infection. And so one of the hallmarks of a Pseudomonas infection can be kind of a greenish yellow pigment or colored compound that's produced, and you can see that there around the wound. We get exposed to this all the time out in the environment, but it generally doesn't cause infections in healthy people, but it can cause problems for people who are diabetic and have weak immune systems, or burn victims are also very likely to wind up with Pseudomonas infections. All right, over on the flip side, we have obligate anaerobes. So an aerobe uses oxygen gas, an anaerobe does not. An obligate anaerobe dies in the presence of oxygen gas. Some good examples are the Clostridium species, and if you guys remember, we talked about the Clostridium genus as being a genus of bacteria that produces endospores. Remember C. diff, Clostridium difficile as an example. And those organisms are also obligate anaerobes. That means that they're not going to be able to survive in the presence of oxygen gas. That's going to severely limit where they can grow in the human body. Most parts of your body are heavily perfused with oxygen gas because your cells need that oxygen gas to be able to do cellular respiration and survive. So there are not very many areas in the human body that are anaerobic environments. Your intestinal tract, though, is. So when you get down in the colon along that inner lining, that's an anaerobic environment. And in fact, the majority of your intestinal bacteria are anaerobes. Or over here, we have a picture of somebody with gangrene, which is caused by Clostridium perfringens. And if you had frostbite or a severe wound and it cuts off the blood supply to some of your tissues, those tissues can become anaerobic because you no longer have blood bringing in oxygen gas. And then if you get exposed to Clostridium perfringens bacteria, they can grow in those anaerobic tissues, and that can cause gangrene, which is not fun. All right, some other categories. This is one you're going to want to know about because you'll hear about this quite a bit. Facultative anaerobes. So a facultative anaerobe, now it sounds like they would still prefer to not be in an oxygen-containing environment, but that's not true. A facultative anaerobe prefers an environment usually with oxygen gas, but it can survive without it. So facultative is telling you what it can do as an alternative lifestyle. And a good example is E. coli, Escherichia coli. That's a facultative anaerobe. It would prefer to have oxygen gas so it can do cellular respiration and make its ATP. But if oxygen gas is not around, it's still capable of surviving in the absence of it. Then you have aerotolerant anaerobes. So these are organisms that don't use oxygen gas for cellular respiration. They have alternative methods, but they can still tolerate it. So if you remember over here, we just said if you are an organism that can survive around oxygen gas, you've got to have a way of destroying all of these nasty oxygen-containing compounds that are going to naturally form inside your cell. If you're an obligate anaerobe, you don't have those mechanisms. So that's why you die in the presence of oxygen gas, because all these free radicals and other types of nasty oxygen-containing compounds build up, and they wind up damaging your DNA and killing you. If you're an aerotolerant anaerobe, you don't need the oxygen gas, but you've still got the mechanisms for inactivating those nasty oxygen-containing compounds. Microaerophiles, you can figure out what that is based on the name. Micro means small, aero refers to oxygen gas, and phile means to love. So they love oxygen gas, but at low levels. So in the atmosphere, 21% of the gases in the atmosphere are oxygen gas. That's too much for a microaerophile. They need oxygen gas at levels around 2 to 10%. Some example pathogens: Heliobacter pylori or Helicobacter pylori. You'll see both of those genus names for this organisms. Or H. pylori. Maybe you've heard of H. pylori before. It's an unusual organism. It's capable of growing along your stomach lining, which is kind of weird if you think about the stomach lining, because hopefully you know that that's a low pH and acidic environment. So most things can't survive there, but Heliobacter pylori can. And then Borrelia burgdorferi, which we talked about that earlier in the class, because that's the spirochete type bacteria that causes Lyme's disease or Lyme disease. And it also is an organism that prefers a lower percentage of oxygen gas for survival. How do we characterize oxygen requirements of organisms in the laboratory? One thing you can do is a test with a special type of liquid broth. And what that liquid broth does is it limits the amount of oxygen that's able to diffuse in from the air in the tube down toward the bottom of the tube. And so what that does is it creates an oxygen concentration gradient in the broth. So your oxygen concentration is high up here and low down here at the bottom. So if you're an obligate aerobe, you're only going to grow at the top of the tube. If you're an obligate anaerobe, you're only going to grow at the bottom. If you're a facultative anaerobe, remember we said those organisms prefer oxygen gas, but they can grow without it. So they're going to grow generally heavier at the top where you have oxygen gas, but they'll still grow elsewhere in the tube because they don't have to have oxygen gas to survive. An aerotolerant anaerobe, remember, doesn't mind oxygen gas but isn't using it. So they're able to grow pretty much at the same level everywhere in the culture. So that's the picture from a textbook. Over here you've got actual oxygen requirement tests being run in a laboratory in these liquid tubes of broth. And here's an obligate aerobe. So you can see you've got a lot of heavy growth, a lot of turbidity up at the top, and the rest of the broth is clear. These are both examples of facultative anaerobes, different species. This one is growing heavily at the top, and it kind of drops off a lot before you get all the way to the bottom of the tube. This one's growing better all the way to the bottom of the tube. So these facultative anaerobes, different species, will have different abilities to grow in low oxygen levels, some slow, some faster. And then over here you actually have an obligate anaerobe. So you can see the anaerobic environment in this tube actually starts about right there. This would be your aerobic part up here. So not growing up here, and it is growing down here toward the bottom. Okay, so in a microbiology laboratory, sometimes you have to culture anaerobes because there are anaerobes like Clostridium perfringens that cause disease, but you can't grow them out in open containers or open incubators in a laboratory because those pieces of equipment are exposed to oxygen gas, and oxygen gas will go in those cells. It gets converted to those nasty oxygen-containing free radicals and other compounds like H2O2, and they don't have any way to inactivate those, so those molecules wind up damaging and destroying those cells. So if you want to grow an anaerobe in the laboratory, a lot of times they're grown in a gas pack like this. So here are some Petri dishes, some cultures that are placed in the pack. You've got a tight seal lid that has an O-ring gasket on it, so it prevents O2 from getting in there. And there's a gas pack in here that emits gases, usually carbon dioxide and hydrogen gas as well. And so that creates, you've got these palladium catalyst pellets over here that react with any O2 in there and limit the oxygen gas that's available. So that creates an anaerobic environment. That's important to know if you're going to work. You might wind up working in a clinic somewhere where, if you're a nurse, you might have to set up cultures, and in some clinics you might have to know if the physician says, hey, let's set this up as an anaerobic culture, you've got to know it has to go into one of these gas pack environments in order for those organisms to actually be grown so that they can be studied. All right, so that finishes up temperature and oxygen requirements. In the next video lecture, that video wound up being a little longer than I wanted it to be, we will move on and discuss some of the other environmental factors that I mentioned. We would discuss as far as the variation that exists out there in the microbial world for their preferences.